Hello all, welcome to another session on pathology. Now we have entered chapter 3, inflammation and repair. Compared to the first and second chapter, it's a bit lengthy chapter. Why? Because we are going to discuss many things. And once you are okay with this chapter, you will understand that the whole pathology is based on this basic chapter. That is why we are considering these chapters as a part of general pathology. Yes, fine. Why? Because the word inflammation is a very common terminology which we come across in our day-to-day clinical practices. What do you mean by inflammation? How the inflammation is happening? Why it is happening? Why the patient experience or complains of symptoms during this phase of inflammation. Inflammation of organs, am I right? Everything. Inflammation, we have different, usually the inflammation terminology ends with the word itis, peritonitis, orchitis, funiculitis. So all this means inflammation. So in this chapter, we are going to discuss things part by part 1, 2, 3, or sometimes if needed, we will go for part 4 also. We will discuss in small amount so that you can understand things very clearly. Fine. Now see, what is actually an inflammation? Whether it is a protective response? Yes, of course. Inflammation is a response of vascularized tissues as a mechanism of host defense to protect our body or from some permanent damage caused by some foreign objects or foreign body, whatever it may be. I'm not mentioning that it is a microorganism or it is a trauma or whatever it may be. So the typical, see the suffix itis, appendicitis, conjunctivitis, meningitis. Am I right? Yes. The typical inflammatory reaction has some steps. It has some sequence of steps. First, what? The whole, I told you in Robbins, always focus on images. Okay? Please open your eyes when you come across any pictures in Robbins. Here, microbes. Here, microbes are going to enter inside our body. How it will enter? It will penetrate through the epithelial barriers. You would have learned in physiology. We have a lot of skin barriers, physiological barriers, immunological barriers. Yes. And once this microbe enters, it has to be properly recognized by our immune system. Our immune system have macrophages, dendritic cell and mast cell. First, it will recognize. And after recognition, it will produce some mediators. These mediators will have some physiological effect. It will produce some physiological responses like recruitment of leukocytes, monocytes, granulocytes. It may activate the plasma protein complement cascades and it may lead to vasodilation, increased vascular permeability, that is the responsible for edema. Finally, all this is done, the microbes has to be eliminated by phagocytosis and the inflammation subsides. Sometimes, if this agent remains in the body, if the body fails to remove these microorganisms within a period of time, this inflammation may progress. So, if it is removed within a particular period of time, we call it as acute inflammation. Suppose if it persists, we call it as chronic inflammation. Very basics. See, first, what is the step? I am just going to highlight the step, recognition. Okay, recognition. How the microorganisms are recognized? We have a mechanism, we will deal in detail. Then recruitment of leukocytes and plasma proteins into the tissues. This is responsible for the vascular permeability, am I right? Cytokines, okay, the complement cascades, the patient may have fever, okay, all those things are there. Then, of course, the stimulus has to be removed by phagocytosis. Fine? Yes. Suppose I told you, if it proceed to chronic inflammation, finally it will cause some repair or some permanent damage that has to be repaired properly. That is we call wound healing, repair, scar formation. Yes, all these are a sequelae of chronic inflammation. Fine? Yes. Okay. So here we are going to discuss what are the components of an inflammatory response, how the inflammatory response starts, who are the people who is going to participate in this from our immune system, okay, how the body is going to respond, how the vascular structures are going to respond. Then finally we will see what are the harmful consequences. Suppose if this inflammation is going to persist for a very long period of time, what will happen to our body? Okay, all those diseases we are going to discuss in this chapter. And before that, let us see a very, very important table. I am going to tell some acute and chronic disorders, and I am going to tell you what are the cells or the molecules which are involved in this injury. This is very important. Why? Because you will get one MCQ question. Okay, you will get one MCQ question in any central institute exams like any set or your PG entrance exam. Okay, all those. See, ARDS, acute respiratory, it is acute. So acute means you can imagine of neutrophils. Okay, asthma, it's like allergy. So allergy means definitely it will be eosinophils. Glomerulonephritis, you will see in chapter 6 immune system. Glomerulonephritis, it is actually due to antigen antibody complex deposition, mainly due to activation of the complement pathways. In septic shock, again we will learn in chapter 4, cytokines are responsible for this. Chronic conditions like arthritis, or I can write tuberculosis, which is a very chronic condition. Here, lymphocyte. Whenever you heard the word chronic, remember lymphocytes and macrophages. Asthma, it may be an acute asthma or a chronic asthma. Chronic asthma, also definitely the one who is going to respond is eosinophils. Atherosclerosis, pulmonary fibrosis, all those we have macrophages and lymphocyte. So what is the gist here? Okay, gist here. Remember, acute neutrophils. And if it is chronic, we have macrophages and lymphocytes. Simple. Okay? Yes. Then we are going to see what are the mediators of inflammation. That is, there we are going to see what are the cytokines, what are the interleukins, what are the chemical substances released by our body, okay, and because of that, what are the symptoms, okay, the patient is experiencing. Then what is acute, what is chronic inflammation. All these we are going to tell. This is a very, very important table, VVIP table. Focus on this. Yes. What are the signs of inflammation? I am not saying symptoms. Signs are the ones what we elicit. Number one, okay, initially there were 4 cardinal signs of inflammation. It's a straight away question. All of the following are the 4 cardinal signs or the cardinal signs of inflammation except. What are they? Rubor, redness, tumor, swelling, calor, heat, dolor, pain. Okay, this was actually put forward by a Roman scientist. His name is Celsus. You should remember his name. Later, Rudolf Virchow. Okay, he's is commonly called as the father of the cellular pathology. Of course, we can say, of course, if somebody asks you who is the father of pathology, you can very boldly tell, of course, Rudolf Virchow. Okay, yeah. He has added a 5th component, that is functio lusa. Functio lusa means loss of function. Loss of function. And there is another scientist, if you go for some quiz competition, they may show their pictures and they will ask you, that is Elie Mechnikov. You would have learned in microbiology also. He discovered the process of phagocytosis. Very important. Okay. What is that? Phagocytosis. Okay. In this paragraph, you have to remember what are the 4 cardinal signs which was put forward by Celsus. What is the 5th sign the Rudolf Virchow has added, that is functio lusa, and what is the contribution of Elie Mechnikov. Fine. Yes. Coming to, okay, see, there are some very basic differences between acute and a chronic inflammation. Acute inflammation will be between minutes to hours. Here it will be for days. Here it will be mainly neutrophils. Here we have told you macrophages and lymphocyte. Here it will be mild, self-limited. Here it will be severe, progressive. So the local and systemic signs will be more prominent during acute acute? This is a very important question. In acute or chronic disease, the symptoms will be more prominent. Answer is acute. Why chronic? Because it's for a very long time. The patient, tuberculosis, the patient won't present with the symptoms of tuberculosis immediately. When he, for example, today I am going to TB ward. Yeah? You have respiratory and TB medicine. I am getting exposed to a TB patient. Whether I will get symptoms like cough with hemoptysis, evening rise of temperature, everything tomorrow? Definitely not. It will take a very long period of time. Sometimes after 5 or 10 years only I will present with the symptoms. So it's a very chronic. So the symptoms may not be prominent. Okay. Causes of inflammations. I'm just going to say the heading. Either infections. Any organisms can cause infection. Any tissue necrosis, myocardial infarction, stroke, radiation exposure, fine, trauma, burn, thermal injury, frostbite, everything. Foreign bodies. Fine. What are foreign bodies? For example, cholesterol deposition, atherosclerosis, gout, uric acid deposition, lipids in obesity. Then immune reactions, hypersensitivity reactions. We have a lot of type 1, type 2, type 3, type 4 hypersensitivity reaction. Any autoimmune reaction, diseases, autoimmune diseases, like you would have learned in physiology, myasthenia gravis. Am I right? Graves' disease. Similarly, we have a lot of what we call it as autoimmune disorders. So, it is a different topic we will discuss in detail in chapter 6. Okay. Now I told you what is the first step. I hope you remember this picture, children. Okay. What is the first step? The microbes, when it enter, it has to be recognized properly. The recognition is the first, it is the initiating step. It is the initiating step in inflammatory reactions. Okay, yes. How these are? There are some receptors in our body which will detect some patterns, which are some proteins which are present in this microorganism, and these receptors are called as toll-like receptors. What are these receptors called as? Toll-like receptors. As of now, I am not going that much detail about this because again in chapter 6, immune system, in immunology or in microbiology, you will discuss about what are various classes, TLR 1, 2, 3, 4, which TLR detect viral proteins, which TLR detect what protozoan or fungal proteins, everything you are going to detail study in immunology. So I am not going that much detail. Then we also have some sensors of cell damage. I told you in apoptosis, okay, whenever there is some injury stimulus, it will be sensed by some BH3 family of proteins. We call them as sensor proteins. Similarly, when a microorganism or whenever, foreign body gets into our body, what will happen? It has to be, it will produce some damage. That damages has to be sensed, and those receptors here we call it as node-like receptor. What we call it as node-like receptors. How I used to remember, suppose a microbe enter inside our body. How our body will respond? Yes, like we will, yes, he is entering. We will nod our head, no? Yeah, that's the way. That is how the body also responds. Yes, so that's why those receptors are called as node-like receptors. Fine? Yes, okay. Then we have other cellular receptors also, and remember I have already told you, please, those who have not listened, please go to my session on what we call as pyroptosis. It is a very highly advanced topic that pathway has not mentioned in Robbins. What is canonical inflammasome and non-canonical inflammasome? Two pathways I have explained there. Interleukin 1 caspase 1 caspase 11 gas-dermin D cleavage. I told you, gas-dermin D has been asked in your AIMS PG entrance exam question. Yes, so that is, please go and refer that session. Okay, yes. So all this, okay, yes. So node-like receptors, toll-like receptors, some other cellular receptors, complement system. All this will help in recognition of the microbes and damaged cells. Microbes are mainly recognized by toll-like receptors. Damaged cell, what is the receptor? Node-like receptors. Okay, yes. See, what are the steps here? Recognition, leukocytes recruitment, removal of the agent, control of the response, finally resolution. Just remember these key points. Okay. Once this is done, we will see about acute inflammation. And acute inflammation has three major components. Number one, increase in blood flow because of dilation of the blood vessels. Blood vessels in the sense small vessels, arterioles, capillaries. As a result of increased blood flow, the increased hydrostatic pressure will be there. So the proteins and all will be pushed up, through the extravascular compartment. We call it as increased permeability. Then the emigration of leukocytes from the microcirculation, that is, the neutrophils will be usually passing in the central pathway of the blood vessels. No, they will come to the periphery. Yes, where the injury is there, they will accumulate there and they will eliminate the offending agent. These are the three important cardinal steps of acute inflammation. As far as your university exam is concerned, acute inflammation is a very standard short form question. How the question will be, as per your new CBME curriculum, they will give a 40-year-old male comes to the surgery OPD. I am writing SX surgery OPD with complaints of pain in the right iliac fossa, okay, radiating to umbilicus, which was acute in onset. What is your probable diagnosis? Whenever you hear the word right iliac fossa, radiating to umbilicus, acute onset, surgery OPD, 40-year-old male, definitely I hope by this time you would have understood the diagnosis. It is a case of acute appendicitis. Okay. Then they will ask you, explain in detail the pathogenesis behind this, or explain in detail the various events in it. That means they want you to explain the steps of acute inflammation. Simple. Okay. Yes, this is your new curriculum. Fine. First, how the blood vessels will react. I told you there will be increase in permeability, then leukocytes recruitment into the site of injury. Here we have two words. One is called as exudate, another one is called as transudate. Exudate is an extravascular fluid. X for X. That means it has high protein concentration and cellular debris. Exudate means inflammation. Transudate, low protein content, no cellular material. Fine. It is an ultrafiltration of blood plasma, and it is produced as a result of some hydrostatic imbalance. There should not be any vascular permeability increase. Two terms, exudate, transudate. Exudate, extracellular fluid, high protein concentration, cellular debris, indicates there is an inflammatory response. Transudate, ultrafiltrate of plasma due to increase in hydrostatic pressure. Simple. Again, transudate we will discuss in chapter shock, hemodynamics. Okay. As a result of this change in vascular flow, okay, then we are going to see how this vascular flow is going to change. Everything we are going in depth. Number one, by vasodilation due to histamine release. First, vasodilation of arterioles occurs, then capillaries occurs. This vasodilation is followed by increased permeability of the microvasculature. So what will happen? The fluid will lose. Red blood cells will start concentrating. Once the fluid loss, blood will become thick. Increased viscosity of the blood. So what will happen? The movement of the blood will become slow. We call it as stasis. See, this is a normal. Normally there will be increased hydrostatic pressure and decreased colloidal osmotic pressure. In exudate, there will be vasodilation, stasis, fluid and protein leakage, indicates inflammation. In transudation, decreased colloidal osmotic pressure. It doesn't means there is an inflammation. That means transudate is not an indicator of an inflammatory response. Why? Because I told you it may be due to increased hydrostatic pressure or decreased colloidal osmotic pressure. Transudate is responsible for formation of edema. What is edema? What is the pathogenesis of edema? Already you would have learned in CVS physiology. We will discuss again in hemodynamics chapter. As far as inflammation is concerned, exudate is important for us. Okay? Yes. Okay, now what happens? Stasis. Initially, the neutrophils normally they are present along the center. They normally travel along the center of the blood vessel. Now the blood become thickened. They starts to settle down along the vascular endothelium. Okay, yes. So this is increased vascular flow. Then comes increased vascular permeability. Everything, each step we are going in depth. How this vascular permeability is increasing? Number one, contraction of endothelial cells. Contraction of endothelial cells. Here comes the question, what is the most common mechanism of vascular leakage? Answer is endothelial cells start contraction. As a result, there will be gap forming between the two endothelial cells. And this will be aggravated by histamine, bradykinin, leukotrienes. Okay, and this response is called as an immediate transient response. Immediate transient response. That means it is immediate, occurs very immediately. Why it is transient? It is short-lived, just around 15 to 30 minutes. This is one response. This is one type of response. What is it? Immediate transient response. It is an MCQ. Q. Put a Q there. It is an MCQ. Immediate transient response is because of answer contraction of endothelial cells. Then suppose in case of some mild injuries like UV radiation, okay, bacterial toxin, this leakage is a little bit delayed. That was for 5 to 15 minutes. It become 2 to 12 hours. And at the same time, the earlier one, it lasts only for a few minutes, short-lived. That is why immediate transient. But here, it occurs after some 12 hours. And it also get what? Long-lived, long-lasting. So that is called delayed prolonged leakage. Delayed prolonged leakage. This is because of endothelial damage. Here comes a controversy. In Robbins, it is mentioned that the delayed prolonged leakage is because of both contraction as well as endothelial damage. But the MCQ books, okay, the authors, they will prefer to go with mild endothelial damage. Contraction for immediate transient response, mild endothelial damage for delayed prolonged leakage. Okay, yes. Okay, what is the best example? Here comes the MCQ question. A patient who has sunburn has which of the following type of response? This is a simple, straight one-liner questions. Answer is what? Delayed prolonged leakage. It is because of what? Endothelial damage. I hope it is clear. See, you can see here there is a retraction of endothelial cells, rapid, short-lived. Okay, and because of endothelial injury, rapid may be long-lived. Okay, you can see the damage. Okay, endothelial is getting damaged, but here see everything is normal. What they are doing? There is only retraction. Only retraction. Here there is damage. Okay, this is long-lasting. This is short-lived. Once this is done, okay, endothelial injury also I have told you already. Okay, yes, in thermal burns, everything, there will be direct endothelial injury. Then, okay, so this is how, okay, this is about the vascular vessels. Similarly, the lymphatic vessels and lymph nodes will also respond. That is not that much important here. Fine, okay. Once vascular reactions are over, what is the next step? Leukocytes has to be recruited to the site of inflammation. How they are being recruited? Already the blood know, they become more viscous. So, ones which are traveling in the center will settle down. Fine? Okay. Okay. And these leukocytes play a very, 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 very important role. Why? Because they are going to perform phagocytosis. Answer is simple. Okay. And how these leukocytes migrate from the center of the blood lumen of the blood vessel to the periphery, how they are going to initiate the further process of inflammation. All this happens by the release of some molecules and cytokines. We call it as chemokines. Let us discuss this process in various phases. Number one, margination, then rolling, adhesion to endothelium, then migration across the endothelium, then migration towards the offending agent or the stimulus. This step they will ask in the form of a flowchart in MCQ. Which of the following is the correct pattern of what? Leukocyte recruitment to the site of inflammation. What is it? Right here, M, margination, rolling, adhesion, migration. Okay, yes, Mrami, or how somehow you remember this. Okay, yes, Mrami. It is margination, rolling, adhesion, migration. Fine, yes. First, we are going to see each of these processes. Each of these processes we are going to see. First one is, what is margination? Margination means the leukocytes present in the center will reach as the periphery. That is what we call it as margination. That is what we call it as margination. Okay, yes. Once margination happens, okay, see, we can go for this picture. Okay, we can go for this picture. Okay. See, you can see here the leukocytes which are present. Actually, the process which is mentioned here, the leukocytes, okay, first undergo margination, it undergo rolling, then finally it has to adhere, that is adhesion, to the endothelium. Then it has to cross the endothelium, that we call it as transmigration. Finally, it has to reach what? Near the offending agents. So these are the various steps. And of course, the rolling and all, there is not that much significant. What we have to focus is how these leukocytes are going to adhere or it attach with that of the endothelium. There are some receptors should be there. There should be some ligands. There should be some chemical molecules which helps, okay, which facilitate these processes. That only we are going to discuss now in details. Okay, fine. Okay. Yes. So this adhesion of the leukocytes to the endothelial cells is mediated by some two group of molecules. They are called as selectins and integrins. Remember, they are expressed on both leukocytes and endothelial cells. Okay, yes. So I told you, what is that? Rolling. So the rolling is mainly mediated by this selectin. Rolling is mediated by what? Selectin. I am writing here. Rolling is mediated by selectins. Okay, yes. And integrins, as the name indicate, integration, that means attachment. They mainly helps in attachment to endothelium. I hope it is clear. Attachment to endothelium. Okay. Remember, children, we have different types of selectin. L-selectin, which is present on L-selectin, which is present on leukocyte, E-selectin, which is present on endothelium, and P-selectin, which is present on platelets. Actually, what are these selectins? They are sialylated. Sialylated means sialic acid attached oligosaccharides which are bound to mucin-like glycoproteins. Not that much important. Leave it. Fine. You have to remember E, L, and P selectins are there. Selectins helps in rolling. That's all. Okay. Yes. All these selectins will be what? These receptors, no? All these will be expressed on this endothelium as well as on our leukocytes, and this is going to facilitate. Okay, this is going to facilitate what we call it as the process of rolling. Okay, just to remember, selectins facilitate rolling. That's all. Nothing going to detail. Okay, yes. And we have something called as Weber palade bodies. I'm not telling about much more about the Weber palade bodies now. Just to remember, it is related to platelets. Okay, because we will see that in hematology in detail. What is Weber palade granules? Okay, where they are. Actually, Weber palade bodies are present in the endothelial cells. Just remember that point. Weber palade bodies are present on the endothelial cells. Okay, and they act as a storehouse of P-selectin. They act as a storehouse of what? P-selectins. Okay, then we have the integrins. Integrins, I told you, it gives the leukocytes to bind or attach with the endothelium. Okay, we have a lot of integrins. Out of that, the most important one I am going to discuss. Number one is VCAM, vascular cell adhesion molecule 1. Number 2, intercellular adhesion molecule 1, and MAC1 and MAC1. Out of that, the most important are VCAM and ICAM. VCAM and ICAM. Okay, what is the main important functions of these integrins? You know, leukocytes are more firmly attached to the endothelium because of these integrins. Okay. Once it binds with the endothelium, what is the next step? It has to cross the endothelium. That is called as transmigration, also called as diapedesis. Transmigration, also called as diapedesis. Remember, the transmigration or diapedesis is done with the help of a molecule, very important, CD31 or PCAM1. Transmigration. What is it? Transmigration is because of, we have learned no what E-selectin, P-selectin, L-selectin helps in rolling. BCAM, ICAM helps in attachment to the endothelium. Similarly, PCAM, PCAM, also CD31. CD means cluster of differentiation. It is actually an IHC marker, immunohistochemistry marker. We have lot of IHC markers in hematology. Okay, of course, in hematology only we will mainly use a lot of IHC markers for diagnosis of various cases. Okay, I will take IHC markers in a different session. At that time, you can bother about that. As of now, just remember CD31 or PCAM1, platelet endothelial cell adhesion molecule. This is the one which is responsible for transmigration. Let us come to this table. Tables in Robbins are very important. L-selectin, E-selectin and P-selectin. How I told you CD31 is for PCAM. CD31 is for PCAM. Similarly, we have an IHC marker for selectin, that is CD62. CD62L, CD62E, CD62P. Already you know where they are present. Don't worry about this. Don't worry about this. Just remember two things. One is ICAM here, another one is VCAM. These are the two important integrins which helps in the process of integration, that is attachment of the leukocytes more firmly to the endothelium. Just remember these 2 points, no other points. Don't waste your time mugging up all those things. They are not going to ask this at any cost anywhere. Fine? Yes, leave it. Okay. So transmigration, PCAM or CD31, this is VIP. Okay, you can't afford to miss this question. Okay, PCAM or CD31, selectins are CD62. I am writing here. Selectins are CD62. CD62, CD cluster of differentiation. It is an IHC marker, immunohistochemistry marker. Okay, I have told you in apoptosis we have annexin 5 as a marker. CD95 is a marker of extrinsic pathway of apoptosis. Annexin 5 is a marker of intrinsic pathway of apoptosis. Similarly, CD62 is a marker of selectin. CD31 is a marker for transmigration or diapedesis. Okay. Now what's the step? It has crossed. The leukocyte should go towards the authentic agent. What is that process called as? Chemotaxis. Chemotaxis. What is that called as? Chemotaxis. Okay. The bacteria will produce some compounds which will attract. They are called as chemoattractants. Okay. Yes. And these chemoattractants will attract the leukocytes. And some of those chemoattractants are interleukin 8. In complement system, we have C5a. Arachidonic acid metabolites like leukotriene B4. I know it is very difficult to remember all those. So what I am going to do, I am going to draw a table here. Okay, 1 second. I am going to draw a table here, and we are going to write down those chemoattractants here. Chemoattractants. All these are MCQ questions. Okay, you may get this. Which of the following are chemoattractants? All of the following are chemoattractants except number 1, leukotriene B4, number 2, C5a. No worries, we are going to discuss all these mediators in detail. Okay, but not that much in detail because you are going to learn this like a huge story in immunology, in microbiology. Okay, so just a gist only I am going to give you. Leukotriene B4, C5a, IL-8, interleukin 8. Okay, anything we have missed? No. Fine. Leukotriene B4, C5a and interleukin 8. MCQ, one question, definite MCQ. These molecules will attract the leukocytes. This will attract the leukocytes. I told you I told you. In acute inflammation, who are the first persons? First line of defense, neutrophils. And these neutrophils will persist only during the 6 to 24 hours. After that, they will be replaced by monocytes. What is the reason? Number one, what they are telling is that neutrophils are more numerous. Fine? Yes. And another thing, it more firmly attached with the endothelium because it is only having P-selectin, E-selectin much more prominent. Expression of P-selectin and E-selectin in neutrophils is very more dominant compared to that of in macrophages. And another thing, what will happen? Why it is replaced by monocyte? Simple. Neutrophils, their half-life is very low. Right? They are very short-lived, so they will undergo apoptosis, they will die within few hours, so immediately the monocytes has to come into action. These are the reasons why we are telling, oh, in acute inflammation, neutrophils, but after a period of time, they are replaced by monocytes. Why they are replaced? Short half-life, they will die. Okay, yes. And here comes a question. In which of the following infection neutrophils persist even after days? What I have told you. I told you that after 24 hours, neutrophils will be replaced by macrophages or monocyte. But in case of Pseudomonas infection, why you are going to tell? Because you are going to study Pseudomonas in microbiology. At that time, you will understand how Pseudomonas or in why Pseudomonas infection, the neutrophils are persisting even for several days. In Pseudomonas infection, the neutrophils will continuously be present for several days. And similarly, in viral infection, neutrophils won't come first. Instead, lymphocytes will come first. These are the two exceptions to the normal rule. Normally, whenever there is an inflammation, first neutrophils will come. 24 hours after, macrophages will come. Pseudomonas, neutrophil will persist for several days. In viral infection, lymphocytes only will come first. Q. You can't afford to miss these points. Open your eyes here. Fine? Yes. Leukocytes are recruited. You can go through all these pictures. Then what is the next step? Yes, what is the next step? This is a picture of myocardium. In myocardial infarction, CVS pathology we will see. First 1 hour, what will be the change in myocardium? Then 1 to 4 hours, what will be the change? 1 to 6 hours, what will be the change? Up to 24 hours, what will be the change? 24 to 48 hours, what will be the change? Okay, see, you can see there will be edema first, neutrophils will dominate, then it will be replaced with macrophages. As of now, don't burn your head focusing on this picture. We will see in detail each hour how the myocardium histopathology. Okay, we will see in CVS pathology. Okay, once this is done, we will see the next step: phagocytosis and clearance of the offending agents. Yeah, the leukocytes have reached the area. It should remove this culprit. How? Phagocytosis. How to do phagocytosis? You are going to tell. Already I have told you in chapter 1, cell unit of health, receptor-mediated endocytosis, phagocytosis. Fine. Then in chapter 2, again I have told you about autophagy. Yes. And now third time I am repeating the same thing. Yes. What are the enzymes? You are going to tell calcium, protein kinase C, phospholipase A2. Okay. All these are the triggers. Let us see the phagocytosis in a bit detail. It is like a story. All these general points, if you just go through these ones, you can write by your own. Phagocytosis, first again, which particle has to be phagocytosed? So first recognition and attachment, engulfment, finally killing. First it will attach, it will engulf. Okay, I will draw a picture. Suppose this is the offending agent. This is our neutrophil. First what it will do? It will go and attach like this. Then what it will do? Okay, it is going to engulf like this. Okay, now here is the microbe. Then finally what it will do? The neutrophil will become normal. It will be completely killed. Okay, death. Fine, death. Yes. Okay, that's happened. How? First, recognition. I told you, not like receptor. Here also there should be some receptors. Just focus on that point only. Just focus on that point. What are the receptors? Number 1, Mannose receptors. Number 2, scavenger receptors. Yes, anybody, can you refresh your knowledge from biochemistry? Scavenger receptors are involved in cholesterol metabolism. I hope you remember cholesterol metabolism. Scavenger receptor SRA, scavenger receptor SRB. These are the various entities. Okay, in Harper it is well in depth. It has been mentioned about the scavenger receptors. Fine. Okay. Then we have some other important receptors that is called as MAC1. Yes, you know Mac, iPad, iPad, Mac, iPhone. Yeah, what is Mac? Yes, MacBook. Yes, that Mac only. CD11, B bar, CD18. These are the two IHC markers. CD31 is for PK. 62 is for 62 selecting. 95 extrinsic marker for apoptosis. Annexin 5 intrinsic marker. Sorry, it's CD95 is a marker of extrinsic pathway of apoptosis, and CD95 is a marker of extrinsic pathway. Annexin 5 is a marker of intrinsic pathway of apoptosis. CD62 selectin. CD31 PCAM. PCAM is involved in diabetes. Super. Okay. Then we have MAC1. What is it? MAC1 or CD11B or CD18. This is the receptor you have to remember, which helps in recognition before phagocytosis. Simple. No other things. Then what is the thing? Engulfment. Who will do this engulfment? Lysosome. Please refer my lecture on autophagy. Please refer my lecture on endocytosis, exocytosis, all those in chapter 1, chapter 2. In depth I have mentioned what is lysosome, what is phagosome, what is autophagosome, lysophagosome. Many terminologies I have discussed. Go and refer there. Okay, yes. And finally, I am going to tell you, yes, engulfment, everything happened. Now, destruction, killing. Okay, now what is happening? Killing happened. Okay, let us take it first. Neutrophils, macrophages, some important properties has been mentioned. Neutrophils, lifespan, days. In tissues, remember, lifespan in tissues is days. When it comes in the blood vessel, it will die within 24 hours. In macrophages, monocytes, we call it in the blood. Macrophages means monocytes in the tissues. Days or weeks. Both are produced from hematopoietic stem cells in the bone marrow. Then, it's a rapid action. It will rapidly produce, or it will rapidly kill the microorganism by producing an enzyme called as NADPH oxidase. Here comes the question. NADPH oxidase is produced by neutrophils, macrophages, eosinophils, basophils. Answer is neutrophils. It causes respiratory burst, increased oxygen consumption. Refer biochemistry. Fine, biochemistry, NADPH oxidase, what is respiratory burst, everything. Of course, we will discuss it again. Then, it will produce very less cytokines. Whereas the major activity of the macrophages is to produce cytokines. Here comes another MCQ question. Which of the following are the major or the dominant producer of cytokines? Answer is macrophages. Then net formation. What is net? Neutrophil extracellular trap, which is a Robbins 10th edition update. What is net? No worries, we will discuss that in detail later. Okay. How we are going to kill? Killing is by reactive oxygen and reactive nitrogen intermediates. You are going to tell me, because already you have discussed in detail about in biochemistry. This is a separate chapter, free radicals. Chapter 2 I have already discussed free radicals. What are antioxidants? What are myeloperoxidase? Which is the most powerful free radical? Yes, can anybody tell you? Can anybody tell me which is the most powerful free radical? Yes, you have option: hydroxy free radicals, peroxyl free radicals, or peroxynitrile free radicals. What is the answer? Answer is hydroxy radical. Okay, this is the most powerful free radical. It's an MCQ question. Fine. Yes, once all this is done, see this toll-like receptor, everything happen, and what is happening? Killing is mainly by ROS. Killing is mainly by ROS. Okay, just focus on this. See, this is recognition, phagocytosis, phagosome, lysosome, finally killing and degradation. All the same picture, same copy. This is a copy picture from chapter 2 as well as chapter 1. Okay. See, NADPH oxidase. This is responsible for production of the free radicals. These free radicals and the most powerful, what to say, enzyme which is responsible for killing the microbes. Okay, you have lot of options. I am giving you option. NADPH oxidase, number 1. Superoxide dismutase, number 2. Catalase, number 3. And number 4 is MPO, myeloperoxidase. Which option you will choose? Yes, MPO only you should choose. Myeloperoxidase is the most powerful killing enzyme. Why? Because it is only going to predominantly produce, what is this called as? Hypochlorite free radicals. They are the most potent killing free radicals. Of course, hydroxy free radicals are there, but as far as myeloperoxidase is concerned, so suppose if the MCQ question is which is the most powerful, of course you can go for hydroxy free radical, because very rarely only in option they will give OCl. But I'm telling that is the reason for which the myeloperoxidase is the most powerful enzyme. Okay, sometimes in AIMS, we have assertion reason type question. Myeloperoxidase is the most powerful killing enzyme. Assertion. What is the reason they are giving? Because it is going to produce hypochlorite free radicals. Assertion and reason are true, and reason is the correct explanation for assertion. Simple. Okay, fine. Reactive oxygen species, hypochlorite. See, myeloperoxidase, NADPH oxidase, respiratory burst. Okay, respiratory burst. Then, so we can say, here comes the question. See, the hydrogen peroxidase myeloperoxide halide system is the most potent bactericidal system of neutrophils. Okay, yes. Then comes, what are the other enzymes? Superoxide dismutase, glutathione peroxidase, catalase, okay, ceruloplasmin, okay, all these are the antioxidants. Already we have discussed in chapter 2. You can refer biochemistry. Already chapter 1, chapter 2, everything many times you have discussed. Okay, yes. There are some disorders which are associated with myeloperoxidase deficiency. There are some disorders which are associated with what catalase, okay, a deficiency. We will discuss in chapter 6, and all those are high yield points. Okay, that is, they are not mentioned in Robbins directly, only they have mentioned some hints. No problem, we will discuss that in short videos. Okay, like 2 minutes, 1 minute videos I will upload. You can watch that, and because that and all you don't expect in those questions in your university exams, but for your PG entrance definitely they will ask only such type of question. We will see short videos and we will learn that easily. Nitric oxide, you have 3 types: endothelial, neuronal, inducible. Okay, you know what are reactive oxygen species, reactive nitrogen species, everything already you know. And you know that lysosomal enzymes, how they are going to kill everything, all these things, you already know. I'm not going to tell anything again. Okay, yes, I'm going to tell only a very important thing, that is nothing else but called as NET, neutrophil extracellular trap. Okay, neutrophil extracellular trap. Actually, what are they? They are a net. They are a fish. For catching fish we will use a net. No, yes. So similarly, the neutrophils will form a fibrillar network which has some antimicrobial substance. This antimicrobial substance coated net will trap the microbe and prevent it from spreading to other areas. I hope it is very simple language I have explained what is neutrophil extracellular trap. For example, these are some microbes. Here we have neutrophils. It is going to form a net like this. Okay, finally trapping this. It will produce some antimicrobial substance. So it can't spread. Simple. Okay, I hope it is clear. Okay. They are produced mainly by neutrophils. It may be asked as a 2-mark question. MCQ. It is a 10th edition update. Of course, in 9th edition it is mentioned, but not this much detail. They are produced by neutrophils. Okay. In response to infectious pathogens, it has a meshwork of nuclear chromatin, which has some antimicrobial peptide and enzyme. Here comes the question. Which amino acid? Okay, which amino acid are involved in this formation of NET. The NET formation will activate an enzyme called arginine deaminase. You have learned in biochemistry. This arginine deaminase is going to convert arginine to citrulline, which leads to chromatin decondensation. Okay, yes. So what are the amino acids? Arginine to citrulline. Okay, arginine to citrulline, arginine, that is responsible for the two amino acids mainly responsible. Arginine deaminase. Okay, yes, these are responsible for chromatin decondensation. Other enzymes, of course, are myeloperoxidase, elastase, but they are not that much important. Here comes the question. Then what will happen? Yes. Okay, so the microbes are there. Neutrophils form a net. It trap the microbes. Fine. And because of this chromatin decondensation, what will happen? The nuclei of the neutrophils are lost. So the neutrophils will die. The neutrophils along with the bacteria, all those stuffs will die. So what will happen? It can't spread. Remember, this net has been detected in blood during sepsis. This has been an MCQ question in a Central Institute exam. Neutrophil extracellular trap are what explained or are found to be involved in the pathogenesis of which of the following disease? Option 1, sepsis. Option 2, inflammatory bowel disease. Option 3, rheumatic heart disease, or somebody will give some other option. Answer is sepsis, septic shock. Okay, yes, the nuclear chromatin. Okay, yes. And another question, it has been, the neutrophil extracellular traps has been also been postulated to be involved in an autoimmune disorder called the SLE, systemic lupus erythematosus. Don't worry, already I have mentioned this in chapter 2 also. This word we will discuss in chapter 6 we will discuss. So what are the two conditions which are associated with the net? Answer 1 is SLE, answer 2 is sepsis. Okay, yes. So that is how neutrophil extracellular. See, you can see this is an the healthy neutrophils. You can see the nucleus are stained red in color. You can see here, and the cytoplasm are stained green in color. And once all the materials, everything is released to form the extracellular trap, okay, what will happen? Finally, the neutrophils will lost their nucleus because of the chromatin decondensation. See, this is a typical picture, children. This picture, the same picture, same pic. Okay, same picture they will give, and they will ask you what is identified neutrophil extracellular trap involved in sepsis and SLE. Very good. Amino acids, arginine, citrulline. Enzyme, arginine deaminase. Okay, these are the key points you should remember. Okay, mainly produced by neutrophils. Okay, yes. Fine, that is about neutrophil extracellular trap. Please focus on the best. Same color, same copy from Robbins. They will give you a question paper, or in your entrance exam, they will ask you to identify. They will give sub-question, which of the following is true, which of the following is false. You can answer everything, no extra points. Remember SLE and sepsis, NET, neutrophil extracellular trap. With this part 1, with this part 1 finish. I am telling you, number 1, NET. Number 2, events of acute inflammation. Essay question. Number 3, what else we have discussed? Yes. Yeah, the leukocyte recruitment. Okay, recruitment of leukocytes recruitment, that may be asked as a SN. SN means short note. Please go through all these points. MCQs, everything, each and everything we have covered. Cardinal signs, Rudolph Virchow, Functio Luisa, Celsus. These are the very important terms. Okay, please go through all of this. Okay, these are all very, it's a must non-topic. You can't afford to miss any of these points. Okay, thank you.